Hello students, welcome to Shore of Sis classes. So in this lecture we will be focusing on the transformation, that is the mechanism of DNA transformation. So first let's start with the definition of transformation. What is mean? So let's start with the definition. What is transformation? It is the change in the genetic constitution of an organism by picking up genes in the remaining of its dead relative. So this is what we called as transformation. Now first direct evidence that DNA transmit genetic information. So DNA transmit genetic information from the experiment of Griffith and Avery. Now Griffith, in the year of 1928, Griffith and Avery, so they discovered there is two strains. First one is the virulent or the smooth type. That is S type we say, that is protected by the polysaccharide capsule, that is having the polysaccharide capsule, and causes pneumonia, and thus it is the pathogenic. So this one is pathogenic and causes pneumonia, and the second type is, so this causes pneumonia, and the second one is the non-virulent type. This non-virulent is the rough type, that is we called as R type also. So this is the one that is without capsule, that is not having the polysaccharide capsule, without capsule, and does not produce any disease. So this is non-pathogenic. So now from the result of Griffith experiments, so the line S type, that is the virulent type, that effect in mice is death. This is the effect, and this one is the form of bacteria. So this one is the form of bacteria. Now the second one, as I told you, is the R type. So this is line R, this is non-virulent type, that affects in mice when this is the effect. So the effect will be the mice will survive. Now when we will give the heat-killed S type, we can see the mice to survive, and in case of heat-kill S type plus line R type, then the mice will be dead. So autopsy of dead mice that shows that they possess both the type of bacteria type of bacteria, that is the R type and the S type in living state. Now, occurrence of living S type is possible only by transformation of R type, formation of R type, rough type to the S type. So this is the only possible way where transformation is occurring, which picks up the trait of the virulence form of dead bacteria. Virulence from dead bacteria. Now, Griffith's work could not prove that what component of the S-type bacteria that causes this virulence. So, component in Griffith model had no proof. So, the Avray and the McCarthy, let me insert a page. Okay, so in the year of 1944, they separated extract of S-type bacteria into the DNA, carbohydrate and proteins. Proteins and carbohydrate. Now the DNA fraction was further divided into two parts. DNA divided into two parts, with DNAs and without DNAs. One is with DNAs, another one is without DNAs. Now these components were then added to separate the culture tube containing the live R type of bacteria, and they result in, first, when the R type bacteria is given with the carbohydrate of S type, then the R type will only form or be activated. And the second one is live R type is given with the protein of S type. First one is the carbohydrate, second one is given with the protein of S type. So in that case, we will also see the rough type. The rough type we will get in the second case also. The third type is when the rough type is given with the DNA with DNAs, then in that case we will get the rough type. And the fourth condition is R type is given with the DNA without DNAs. DNA without DNAs. So in that case, both type of bacteria will form is R type and S type. So these confirm that this trait or the genes of virulence is located in DNA, and DNA is a genetic material. So from these experiments we get that DNA is a genetic material. So thank you for watching this video.